We used to have final exam for the course, but because of COVID, everything is online this year. We will not have a final exam. I want to still show you some things for final preparation. First, there is a lecture I want the course to watch. It's on the Canvas website. The lecture is about reproducible research, how to make the data and the code public so others can reproduce the result. But that video is really, when data is available, other scientists discovered that the analysis has been fabricated. And so we need to be very, very careful. Data fabrication is really a betrayal of your career. And also patients could be hurt. So we have to be very, very careful to make sure we're doing the right things and, by no means, never, fudge the data or the analysis. So I want every student to watch that lecture. It's really entertaining and also educational. And so this year we do not have the final exam, and grading will just be based on the 6 homework. And we would appreciate the students to fill out the course evaluation and also even the last survey after you submit the homework 6. Please help us improve the course. We do try to take the students' feedback carefully and adjust the homework. If some questions are too hard and the students are complaining about some lectures, we do try to improve. And we try to make the homework better every year and try to make it around the same hours of effort for each homework evenly, even though, I think every year we find some surprises here and there. So this year, some students complain that we have a lot of online pre-recorded lectures. I hope you could see that a lot of the pre-recorded lectures happen more earlier in the semester because those contents have not really changed so much. These techniques are 10 years in the making, so we didn't really have to improve on the teaching too much, but a lot of later lectures, such as single-cell RNA-seq, single-cell attack-seq, cancer immunology, or CRISPR screens, these things are still changing so much, and so we really want to have live lectures so you can see the recent progress and the recent results in this field. So normally, if we have the final exam, we would ask students to answer different type of questions. So I want to just at least give students a taste of what the final exam look like. And so we can use this time to poll the students here. There will be usually some true or false questions. So let me ask, multidimensional scaling can perfectly capture the pairwise distance between nodes. Is it true or false? Can students enter your solutions or your answers in the chat window? Is it true or false? Can multi-dimensions scaling perfectly capture the pairwise distance between the nodes? The answer is false. Depending on your total dimensionality, MDS try to capture as much of the pairwise distance as possible. But if you only give it a single dimension, it will just have to do what it can. The more dimension you have, the better it captures the final dimensionality. But it by no way perfectly capture the pairwise distance. Second question, BAM file is usually used to store the coordinates of the genomic intervals. Is it true or false? The answer is actually false. BAM file is usually used to store the mapping of the reads. If you just want the coordinates of the intervals, you use a BED file. That's a much smaller file. BAM file usually is really big and you just store the locations of the read. The third is epigenetic information is encoded in the DNA sequence. Okay, this one is easier. I guess the definition of epigenetics is the information, is inherited information not encoded in the DNA sequence. And the last one is a gene can be an oncogene in some cancers, but a tumor suppressor in other cancers. Is it true? Yeah, this is true. Yeah, many genes can be oncogenes in some cancers, but tumor suppressors in other cancers, sometimes depending on the mutation pattern. But those genes are, I guess, just really important in terms of gene regulation. They can't be too high, can't be too low. Either way would be bad. Okay, so next one. Usually in final exams, we also have multiple choice questions. So we want to ask, which of the following linkage consider pairwise distance of nodes in hierarchical clustering? Can people enter your solutions? Which of the following linkage considers pairwise distance of nodes in the hierarchical clustering? The answer is D, average linkage. Complete linkage is the maximum. Single linkage is the minimum. The pairwise, I guess, well, yeah, so average linkage is using the average pairwise distance. Single linkage is using the minimum distance of the, I guess, yeah, sorry, maybe this question is not as clear. A, C, D, there's no median linkage. The complete linkage, look at all the pairwise, but take the maximum. The single linkage is considering the pairwise distance, but using the minimum. Average is doing the average of all the pairwise distances. Okay, let's try another multiple choice question. Compared to SmartSeq, droplet-based single-cell RNA-seq method has lower dropout rate, lower profiling cost per cell, use UMI to separate transcript in different cells, and is more accurate transcript readout per cell. Which one is correct here? This is a multiple choice. There are multiple answers, multiple correct answers in here. Droplet-based single-cell RNA-seq is, it costs less per cell. The whole experiment may or may not be cheaper than the SmartSeq. But SmartSeq, the cost depends on how many cells you have to prepare. Per cell is more expensive. Droplet, the whole experiment cost about the same amount, but per cell is less. And droplet-based approach for RNA-seq, they do use UMI to separate the transcript in different cells. But SmartSeq has lower dropout, so is more accurate for in terms of per cell, the transcription expression level is more accurate in SmartSeq. Okay, so droplet is B and C, and SmartSeq is A and D. So next one is short answers. If you use hidden Markov model to predict a protein secondary structure from amino acid sequence, what are the parameters in this hidden Markov model, and what do they mean in this scenario? So what are the parameters in here? So hidden Markov model has initial probability. What is the initial probability in here? The initial probability in a hidden Markov model for protein structure prediction is, in general, if you look at protein structure, so protein structure prediction problem, the observations are amino acid. The hidden states are whether they are alpha helix, beta strand, or loops. And we're trying to predict the secondary structure based on the amino acid sequence. And the initial probability is, how often does the protein start with a loop or something, like so the starting point of the three structures. What are the transition probabilities? The transition probability is what is the likelihood that a alpha helix will follow the next, if one amino acid is an alpha helix, what is the chance the next amino acid again is an alpha helix? Or beta strand to beta strand, loop to a loop, and also alpha helix to a loop, alpha helix to a beta strand, and so on, right? So that's your transition probability. What is the initial probability? The emission probability is, if you are alpha helix, what's the likelihood you are able to see amino acid, valine, glutamine, phenylalanine, and so on? If you are in a beta strand, what is the chance you are seeing a isoleucine or just different amino acid, and so on? And so that's kind of how you use hidden Markov model to predict protein secondary structure prediction. So the next question is, if we have this table in here of comparing two groups, and we're doing hypothesis testing, right? And so what is the sensitivity? Can you write your answers directly into the chat window? If you have to write, x plus y divided by z or things like this, what is sensitivity? Yeah, this is the right answer. Sensitivity is if these two genes are if a gene is indeed differentially expressed, it is like two groups different. Let's just think about a differential expression. Sensitivity is if the gene is indeed differentially expressed, what's the chance you're going to call it? And so if the gene is differentially expressed is M1, and you're able to call it is S, so S divided by M1. What's true negative rate? True negative rate. So these are, the two groups are not differentially expressed, and you are not calling them. So it's U divided by M0, exactly. What is false discovery rate? What is false discovery rate? False discovery rate is among all the differential genes you called, which is R. What are the false positives, which is V? The V means the two groups are similar, but you call them different. So it's V divided by R. That's false discovery rate. Okay. What is the positive predicted value? Positive predicted value. Yeah, it's S divided R by R, exactly. So if you call it to be different, what is the probability that indeed they are different? So S divided by R. Okay, so I think we have lab notes on this. You can go back and check this. All right. And then there are usually some long answers. So throughout the semester, we taught you many different algorithms, and we want to make sure through these long answer questions that when you really deal with a biological data with some high throughput data, you understand how you string these different algorithms to analyze the data and generate hypotheses and get a paper published. And so this is really a way for you to see how these different methods are connected. So let's look at this scenario. In the tumor profiling study, your collaborator conducted RNA-seq of 50 breast tumors, and they're matched nearby normal tissues. And there are also some basic clinical information, such as patient age, tumor stage, survival, and there's no treatment. These patients haven't been to any treatment information. And so the preliminary analysis of the RNA-seq found that for many cases, samples from the same patients are clustered together instead of cancer versus normal clustering into two distinct clusters. So ideally, you want the cancer patients are together, all the normals to be together, or maybe the normals together, the cancers are at least in some subtypes. But in this case, you see a lot of same patients clustered together. Describe how you would like to analyze this data starting from FASTQ. And after the initial QC and normalization, are there any potential downstream analysis you could do to help your collaborator? Yes, so clearly this data, well, not clearly, but based on what we've seen, it seemed to have some batch effect, right? But since this is a long question, you say, okay, let me start from the beginning. We look at the data together. You can imagine you do some star alignment to map the reads, right? You do RCQC to make sure all the samples are correct. Like a QC is okay. You can use HT-seq. By the way, here there's no single correct answer. You can also imagine using salmon to get to the expression index, right, to get the TPM. And then you can use either hierarchical clustering or PCA to cluster all the samples to really take a look, whether there are some outlier bad quality samples or whether there are some batches, right? And so you want to find out whether these are different batches in terms of processing, in terms of whether they're coming from different hospitals or things like that. You want to remove the batch effect from the RNA-seq. And then you cluster them again and see whether the batch effect has been removed. And after the batch effect removal, you might want to see whether there are subtypes in the tumor. So the ideal clustering is normal samples are together. And the tumor samples could be just one cluster or could be, 2, 3 different clusters. For example, if you were to compare the TCGA breast cancer, the normals should be together, but the cancers are divided into luminal, HER2, and the basal breast cancer. And then you want to calculate the differential expression between the tumor and normal, or also between different subtypes. You want to see, what's the difference between a HER2 versus luminal breast cancer. You see, luminal is high in ER and HER2 is high in HER2 expression. You can also look at their differential expression compared to the normal. And based on the differential expression, you can run either David or gene site enrichment, like a GSEA, to look at, gene ontology or pathways that are upregulated in cancer versus normal or in different subtypes. Because this is tumor sample, you can also kind of try other things. You want to see, do they have different immune infiltration? You can try immune decount, right? 6 different immune infiltration methods you ask. Does the tumor has more macrophage or more T cells? Do they show difference? And then finally, you want to ask, also you want to see maybe gender. Do men have more T cells than females, or things like that, right? You can also try things like TRUST to look at immune repertoires to see, whether the immune repertoire is related to immune cell infiltration, is related to some signaling. You can try a single sample GSEA to look at the immune signatures in the single samples, or it doesn't have to be immune signature. It could be any signature you want to see, evaluate them. And also you want to look at Cox regression to see whether the tumor subtypes, immune infiltration, do they have, or gender, or, you know, for lung cancer, you might also want to collect smoking status or age. Do you see difference of immune infiltration associating with age? Also tumor stage, and do they have effect on survival, adjusting for patient stage and tumor stage. Okay, so that's some ideas of how you would go about analyzing this data. Let's try another scenario. In the K562, this is a cancer cell line. The ENCODE consortium has generated ChIP-seq data over 100 different transcription factors and tens of different histomarks, as well as RNA-seq, bisulfite sequencing, DNAs, also ATAC-seq, and these have been pre-processed to pass QC. So you don't need to worry about QCing ENCODE data. These have all been QC'd. And so recently, a group discovered a brand new histomark. Let's just call them H2XYZ. I'm making it up. It's a new histomark, right? Oh, great, epigenetics. And they have generated antibodies, so now they can do the first H2XYZ ChIP-seq in K562. There is no perturbation. Let's just make it simple. Can you help her conduct the initial QC of this data to better understand what this new histomark, to just characterize this new histomark? What would you do? So you start from FASTQ file of this H2XYZ with some input control FASTQ files. What do you do first? So you can see here, this is similar to the other one. You start with QC, right? You want to do fast QC, you do the read mapping, you make sure it's not too redundant. You use max to call the peaks, and you also want to check the signal to noise in your ChIP-seq. You want to check the distribution of the peaks across the genome. You want to see, is my histomarks enriched in the promoters, exons, introns, or intergenic regions, right? Things like that. You might want to compare. You check the peak overlap with of the X2, XYZ with regulatory sequence, because there's like a union of DNAs. How good is that overlap? Does it happen in the enhancer regions, right? You can look at evolutionary conservation, and also you compare with other ChIP-seq using System Toolkit to ask, what other ChIP-seq has overlap with my ChIP-seq of interest? Does XYZ look like K27 acetylation? Does it look like DNA methylation? Does it look like estrogen receptor binding? Or does it look like some other, published ChIP-seq data? And also you can use, you can assign the reads of the XYZ to the nearby genes, and you can look at the putative target genes of XYZ. And if you have differential expression, if you have gene expression, this is similar to, remember your homework 4, when you were trying to look at DNA methylation. You want to see whether DNA methylation is associated with active gene expression or silent gene expression. You want to see whether the mark is appearing in the genes that are silent or active in the cell. And so at least I think that will give you some idea, so in this compare, actually, we're already using some external data in here, but you can imagine you integrate the ChIP-seq with the expression data with all the other external data out there. Okay, so that's roughly how you would probably kind of characterize a new histomark. All right, let's look at another scenario. So there are 12 different cell lines of cancer type X with mutation on gene A and 6 without mutation on gene A. So this is all of the same cancer type, but 6 with a mutation and 6 on wild type of this gene. And so you have generated CRISPR screen. Oh, and RNA-seq and attack-seq of these 12 cell lines. How do you analyze that data? I would say it's always the same. You start with QC and process each data type. CRISPR screens by itself, RNA-seq by itself, attack-seq by itself, right? So initially, you do your QC, you do your batch effect removal to make sure the data is usable. The second is to compare the wild type versus mutant cell lines. In terms of RNA-seq, are there differential expression? In terms of attack-seq, you compare the wild type with the mutation. The CRISPR screens, you compare the wild type with the mutation. So second is usually compare. The third is integrate. How do I combine the different modalities? How do I integrate the RNA-seq and attack-seq of the same cell line? For example, through System Go, you ask, are the genes that have more attack-seq peaks and also higher RNA-seq, are they more essential in the CRISPR screens, right? So you start to kind of look at your data that's kind of more integrated. And finally is to check the external data. If there are some genes that become more essential, if there are some pathways that are becoming more essential, you look at, say, TCGA, other public data to see what is the disease or clinical relevance of what you are finding in this 12 different cell lines, right? And so the last scenario I want you to kind of go back and think again, same thing. So we have 10 patients with immunotherapy, 5 responders, 5 non-responders, and you have generated single cell RNA-seq of pre and post-treatment tumors. So 10 patients each before and after immunotherapy treatment. And so again, you first want to do your standard QC, and then you want to do the comparison before, after treatment, responder, non-responders. You want to look at their immune cell status, immune cell infiltration level, differential expression, and things like that, right? And then, but by the way, once you do differential expression of the different cell types, you can do gene ontology, pathway enrichment, and the third is to check other cohorts, check immunotherapy, see whether other immunotherapy, to see whatever you are seeing is reproducible in, or at least it's a phenomenon holds in other cohorts. Okay, so that's kind of what the final exam should look like. Hopefully this will kind of help you bring all the first 6 homework together. Okay, I think, sorry.